In this adorable noon, I, Aisha Malik, Assistant Professor, Faculty of Life Science Department, Mandsaur University, welcomes you all for attending this prestigious session entitled Enhancing Nitrogen Fixing Biofertilizer Through Diazotrophic Biofilms and Synthetic Biology. Feedback link will be provided at the end of the session in the chat box. Kindly fill it correctly for getting e-certificate. Now, I would like to invite Dr. Shekhar Jain, HOD, Faculty of Life Science Department, to introduce our key speaker, Dr. George.
Thank you very much. I hope you can all hear me. I will now start sharing my screen and start the talk. Hello, everyone.
1 minute, George.
Yes?
Yeah.
Yes.
Thanks, Ayushi. So, dear participants, today we are going to organize one more webinar. In the series of webinar, today we are going to discuss about the very vital component of the living system, that is nitrogen. So, nitrogen is not only important to the plant for the human beings also, or we can say for the all living beings. Nitrogen is a vital part of important part of the DNA and protein. It is just an example. We cannot imagine life without nitrogen, and we all know the 78% nitrogen present in the environment is useless. We can't use it directly until unless we will fix it. So today we are having one of the renowned expert in the field of synthetic biology, who is working on the nitrogen fixing bacteria since last 25 years, and he has achieved too many milestones in the field of nitrogen fixation. So today we are having Dr. Jörg Schumacher from Imperial College of London. He has expertise in nitrogen fixation. His team is working continuously for the improvement of biofertilizer in terms of nitrogen fixation, so that the nitrogen crisis, which is one of the important problem that the world is facing, we can ameliorate this problem. So today, I am very privileged to announce that we are having Dr. Schumacher. He has accepted our invitation to deliver a lecture on nitrogen fixing bacteria and how to improve the biofertilization. Today he will discuss about the nitrogen crisis and how we can overcome this problem by using nitrogen fixing bacteria. Along with this, he will also discuss the recent findings. What he, as I told, that his team is continuously working on nitrogen fixation. So recently, what a finding he got in field of nitrogen fixation, he will share with us. With this note, I would like to invite Dr. Schumacher. Please elaborate us about the nitrogen fixation. Please, Dr. George.
Thank you very much, Shekhar, for this nice introduction. Yes, I will now share my screen. Is that how we're going to proceed, Shekhar?
Yeah, please. Yeah, share.
Currently not. Okay.
Now it's visible.
Can you see the slides now? Can you see the slides now?
Yeah. Yes.
Okay. So hello, everyone. I'm very pleased to be with you online in these strange times. So my talk is entitled Enhancing Nitrogen Fixing Biofertilizer through Diazotrophic Biofilms and Synthetic Biology. I will start this talk by outlining a little bit the problems associated with the production and application of nitrogen fertilizers, of chemical nitrogen fertilizers in agriculture. I will then talk you through our synthetic biology approaches, which we've been conducting for several years now, in order to enhance the nitrogen fixing capacity of plant-associative diazotrophs. And I will then talk about a recent discovery we've made, which I think is of very great importance in the field of nitrogen fixation and biofertilization. So, again, I'll talk about the nitrogen crisis first and possible biotech solutions. Then I'll introduce a little bit the state of the art and talk about current challenges and how we try to overcome them. Now, we all know the world population is growing and we need to feed them. Now, to do so is, if you remember the Green Revolution, which has increased, which has applied high yielding crops as well as chemical fertilizers and the use of machinery and the use of pesticide in form of agrochemicals has, of course, largely enhanced the productivity of agriculture. But the increased application of nitrogen biofertilizers since the 60s, say, correlates very closely with the population growth. And it's estimated that about 30 to 50% of the world population depends on the application of chemical fertilizers. Now, chemical fertilizers are produced by the Haber-Bosch process, which reduces atmospheric dinitrogen into ammonium. This process is highly energy costly. It's because it has to be done at very high temperatures as well as high pressures. And it consumes about 2 to 3% of the world's fossil energy. So there's one downside to this chemical fertilizer. The other one is that the excess nitrogen fertilization, especially in the developed countries, runs off into the aquatic systems and resulting in eutrophication and algal blooms. This then leads to what we call coastal dead zones, and they're marked here in red spots. So you can see that the over-fertilization of the coastal seawaters suffer from this eutrophication, where in the end, if the ecosystem turns over, no fish can live anymore. There's another problem. Due to the nitrogen fertilization of agricultural soils, the production due to denitrification leads to increases in N2 emissions. And that is increasingly seen as a major problem. Overall, the global nitrogen imbalances have been deemed as the second biggest ecological challenge for human development. And I have a short video here from the OECD, which I'd like to share, who makes those downsides of biological nitrogen, of chemical nitrogen fertilization. So there's a YouTube one. Share that.
There are 5 main impacts. Nitrogen pollution is linked to the formation of particulate matter, and that particulate is one of the main ingredients of air pollution and urban smog. The concentrations of particulate in air exceed World Health Organization standards in many major cities around the world, and the human health costs of that outdoor air pollution alone are enormous. They've been estimated at about $1.9 trillion in OECD economies and over $3 trillion in the large emerging economies. That is a problem so large it cannot be ignored. Nitrogen pollution in water bodies can cause the excessive growth of plants and algae. They create dead zones where fish and other aquatic species are unable to survive. There have been 880 ocean dead zones identified, and the number is rising. The soil pollution, excess nitrogen can make soil more acidic, and it may damage the microbial communities that effectively give soil its life-supporting capacity. We know that nitrogen pollution can lead to a loss of biodiversity, both in terrestrial and aquatic environments. That's caused by nitrogen-loving plants outcompeting other species. Nitrous oxide is a powerful greenhouse gas, responsible for about 6% of human-induced warming to date. It also damages the ozone layer, and it's the ozone layer that protects life on Earth by absorbing some of the harmful ultraviolet radiation coming from the sun.
Okay, so we do have a problem. So what are the possible biotechnology solutions or other solutions? Well, the easiest would be if we would all become vegetarian. Oh,
Nitrogen pollution.
sorry about that. So much of the nitrogen overuse is to produce meat, in essence. And so that so many Indians are vegetarian, I would like to congratulate you to that. And if we were all to become vegetarian, much of the nitrogen crisis would not exist. Here you see one of my breakfasts when I was over in India about a year and a half ago. Now, oops. Okay. So what are the biotechnology solutions to the nitrogen crisis? Let's first think about the ecological systems where biological nitrogen fixation takes place. And biological nitrogen fixation can, in principle, replace chemical nitrogen fixation. So nitrogen fixation is, of course, only done by bacteria, by specialized bacteria that have the nitrogenase enzyme. There are different ecologies to this. You probably all know about the symbiotic nitrogen fixation that happens between rhizobia and legumes. In those, the bacteria live in what we call root nodules within the root, fixing nitrogen into ammonium and then providing nitrogen compounds to the plant in return for carbon sources from the plant. This is supposed to be the most efficient nitrogen fixation ecology. The second one is where nitrogen fixing bacteria associate to the roots of the plant or live in the rhizosphere, again, where the plant produces carbon sources from photosynthets and in return receive some of the nitrogen those nitrogen fixing bacteria produce. In the last case, we have the completely free living nitrogen fixing bacteria, which are called diazotrophs, that produce nitrogen and can in principle also to some degree enrich the nitrogen content of the soils. So what are the main biotech strategy to enhance biological nitrogen fixation? The first one, which is carried out a lot in India, for instance, or in South America, is to isolate the best nitrogen fixing bacteria from the soil to test them for their nitrogen fixing capacity and other plant growth promoting traits and use them in the plant system. That can, for instance, be done by coating the seeds with those beneficial bacteria and then before planting. And I'll talk about that later in my talk as well. Other biotech solutions is to engineer symbiotic nitrogen fixation in non-legumes. This is done in laboratories of Giles Oldroyd, for example, in the UK, where they try to engineer the plants in a way that they would be able to induce the differentiation of the root system to nodulate and have, just like we have with legumes, have a symbiotic nitrogen fixation. But this is rather a very long-term goal. It's not going to happen over the next 10 years, I believe. Then there's a very interesting other type of research in biotechnology, which tries to transfer nitrogen fixation capacity into plants. Probably the leader in the field is Luis Rubio in Madrid, and they've made some good progress. So they try to transfer the nitrogen fixation genes into the mitochondria of plants, especially in the leaves. Now, what they have shown so far in the model yeast, that they can transfer some of the nitrogen fixing genes into the mitochondria, and these are functional. This is particularly a step forward because the nitrogenase is highly oxygen sensitive, and the mitochondria, they hypothesize, would provide an environment that is both rich in energy, because nitrogen fixation takes a lot of energy in form of ATP, but also be low in oxygen so that the nitrogenase can function. So some of these parts are being now assembled still in yeast because plant mitochondria can so far not be transformed with novel DNA elements like the NIF genes. So there's a roadblock there, but so far they've at least shown that when you express the nitrogenase components in the mitochondria, that these proteins are functional. And there are also attempts to improve the nitrogenase function itself, because the nitrogenase is a rather slow protein. It's not very efficient. So if we can enhance the catalysis of the nitrogenase, that would help. Others, like at the MIT in the US, they're trying to optimize the NIF cluster for better expression. But all of these haven't really been shown to actually improve biological nitrogen fixation, but it's certainly very strong ongoing work. And what we've been trying to do is enhancing the biological nitrogen fixation capacity for biofertilization. And when we started out with this work, we started from the very simple premise that diazotrophic bacteria, their main nitrogen metabolism consists of two parts. It's nitrogen fixation itself, so taking atmospheric dinitrogen and reducing it to ammonia and ammonium, which is then further assimilated into other nitrogen compounds via the nitrogen assimilation pathway, which basically goes from alpha ketoglutarate from the Krebs cycle, then into glutamate and then glutamine, from which all other nitrogen compounds derive. So nitrogen fixation is the ammonium production process, while as nitrogen assimilation, the ammonium consumption. So with regards to root-associated diazotrophs, you would get carbon sources in form of sugars, say, which would power the nitrogen fixation activity. Now, it's long been known that if you either reduce the nitrogen consumption with regards to nitrogen assimilation of the bacteria, or if you increase the nitrogen fixation, diazotrophic bacteria will excrete ammonium. And in principle, this ammonium can then be used to better fertilize the root systems. And this has been shown in a number of bacteria, Azotobacter, Klebsiella, and a number of other diazotrophs. Now, the issue with that is that if you reduce the nitrogen assimilation pathway, is that the bacteria don't grow very well because they're basically nitrogen starved. So one study I would like to highlight is from a Brazil group. What they showed with Azospirillum brasilense, which is often used for biofertilization, is that a mutant in nitrogen assimilation pathway resulted in very, very strong nitrogen-dependent growth promotion. You can see that here on the left, you have a highly fertilized, chemical fertilized plant. Then further to the right, you see a number of controls with other bacteria, diazotrophic bacteria. But when you look at the one on the right, which has very high growth promotion under low nitrogen fertilizer, it has about 70% biomass compared to the one that has been fertilized chemically. And this, I find it's really encouraging. It's an excellent study. They've traced the nitrogen from the nitrogen into the plant tissue and quantified that, and it really looks good. What you have to keep in mind is under laboratory conditions, but it shows in principle this could work. So in order to make the diazotrophs produce and excrete more ammonium, we have to provide an oxygen-free environment for nitrogenous function. Then we thought we would make diazotrophic nitrogen biofertilizers fit in soil, because obviously in the soil you have competition with other bacteria. And there are, of course, regulatory barriers, at least when you think of genetically modified bacteria, in most countries not allowed to be used in agriculture. But I won't talk about the regulatory barriers for the moment. So our idea was quite simple. In order to make diazotrophs produce and excrete more ammonium, if we had a system where we could tune nitrogen fixation as well as nitrogen assimilation, we could optimize this balance between those two processes in such a way that we both excrete ammonium on the one hand. On the other hand, we would not penalize the bacteria to suffer so much from overproducing ammonium so that they would not grow. So first we have to look at how these things hang together, nitrogen assimilation and nitrogen fixation. So nitrogen fixation produces ammonium, and as I said earlier, it goes via alpha-ketoglutarate into glutamate and glutamine. And this is catalyzed by the glutamate dehydrogenase as well as the glutamine synthetase. And glutamine and alpha-ketoglutarate together, their balance, so glutamine concentration in the cell over the alpha-ketoglutarate, describes what is the nitrogen status of the cell. And this is in Klebsiella oxytoca, the model. It's similar in other diazotrophs, but there are variations to the scheme. So glutamine signals nitrogen sufficiency. Then you have a pathway over P2 and Glyn-K proteins, which impact on the expression of NTRB and NTRC. NTRB and NTRC are, especially NTRC is a two-component system, and NTRC is a transcriptional activator of nitrogen assimilation genes. There's 48 of them. as well as NIF-L and NIF-A. NIF-L and NIF-A are themselves transcriptional regulators of the NIF genes, which are about 20 of them. So the problem here really is when cells like bacteria like Klebsiella oxytoca, when they have produced enough ammonium for their own use, they usually shut down nitrogen fixation. And that is not something, obviously, we want. We want them to overproduce surplus ammonium. So here is the regulatory cascade again, NTRB and NTRC. NTRC gets phosphorylated, switches on the regulator of NIF genes as well as nitrogen assimilation genes, which then results in NIF transcription. And both NTRC and NIF-A are sigma 54 activators. Now, sigma 54 is the main alternative sigma factor in bacteria, or it's also the only one. You are probably more aware with the sigma 70 system, where the RNA polymerase binds to where you have the promoter sequences as -10 and -35 with respect to the transcriptional start site. Sigma 54 is different. It binds to -12 and -24. What is also different is that activation of the RNA polymerase is regulated in a completely different way, where the activator like NTRB, NTRC or NIF-A bind upstream of the DNA, and then it forms a loop to contact the sigma 54 RNA polymerase, and then ATP hydrolysis is needed to form the open complex and transcription can occur. So knowledge of this, we thought what we could do, we could produce or design and hopefully produce functional chimeric proteins whereby we would use our knowledge of those sigma 54 activators like NTRC and NIF-A in order to swap domains between different activators of the sigma 54, so that then we could rewire the cellular signaling process to something we want to design. I won't go into the different technical problems in this protein engineering approach, but the principle is illustrated here. So if you have low ammonium outside the cell, NTRB and NTRC will ultimately then kick in nitrogen assimilation. So what we did is we used a different sigma 54 activator, PSPF, and made a chimera of the catalytic domain of the PSPF system, which is regulated by a membrane stress in this case, and then have the helix-turn helix DNA binding domain of NTRC put onto this chimera, and then making it functional in order to activate the transcription of nitrogen assimilation. Then we get rid of NTRC by a knockout, and then we tested if this works. Again, these are just on the left-hand side. Here is a number of controls using different mutants and different systems. So basically what we showed here in E. coli is that we can activate transcription of nitrogen assimilation genes via two ways. So with our chimera in the cell, we can either induce transcription via IPTG, which largely just increases the number of the transcription factor, of the chimeric transcription factor. But more importantly, we could also increase transcription via this reporter system here by using arabinose, which activates, or which is a signal to activate the PSPF moiety of our chimeric protein, and therefore we can tune transcription of nitrogen assimilation genes. Here on the left-hand side, you see RNA-seq data, and here you have nitrogen assimilation genes. There are about 48 of them. And what you can largely see that under nitrogen-rich conditions, only few of these genes are strongly transcribed when you compare that under nitrogen-poor conditions. And what we could show that with the chimera, we could, to a good degree, boost the transcription of the nitrogen assimilation genes using our chimeric system once we induce it. And when you compare with the copy numbers, so what we do here, we look at the RNA-seq signals for each individual genes in the nitrogen assimilation genes, and divide it by the number of the transcription factor we measure directly in the cell. So you have an expression strength of a certain gene, and you divide it by the number of synthetic transcription factor we produced. It resembles pretty much what we've seen here under the nitrogen poor conditions in the wild type cells. So some of those genes are super activated, and some of them less so. So by and large, I think those synthetic transcription factors allow to mimic or force, if you like, the same transcriptional response to a large degree compared with the wild type system. But we can do that now under nitrogen-rich conditions. So as I said initially, the idea is that if we can have this nitrogen assimilation, nitrogen fixation tuned on while being nitrogen-rich in the environment, we should be able to have a stable overproduction of ammonium, which would then be released to the outside. And this is in part shown here. So here you have in the wild type situation where you have the nitrogen status of the cell, again given by glutamine over ketoglutarate, we can induce that, or we can increase that ratio using our synthetic transcription system. So that's all very nice. And I will come now, this is still under development, and hopefully we will get the system as initially designed and then produce better nitrogen fixers. Now, the other challenge is, of course, to provide an oxygen-free environment for nitrogenous function. The oxygen concentration in the soils is often believed to be too high for nitrogen fixation. And most studies in nitrogen fixation have, of course, been done in the lab, in batch cultures. And it was thought that most of the plant-associated diazotrophs wouldn't be able to fix nitrogen in the wild because of the oxygen problem. Now, when we were still working in E. coli a few years, well, 2.5 years back, we made this very strange discovery. So what we were doing is we were producing reporter plasmids so we can measure transcription of NIF genes. Here, for example, NIF-H, LAC-Z plus XGAL system, or doing a slightly different one, but basically a fluorescent reporter system. Now, when we were cloning these plasmids in E. coli, we oddly found in the presence of NIF-A, so we transformed E. coli with NIF-A, the transcriptional activator of NIF genes, and then when we were testing out those reporter constructs in E. coli, we saw that NIF genes were transcribed. And that was odd. So if we have a system where we have NIF gene transcription in E. coli, when we stroke them out, we thought that wasn't possible because of the oxygen situation. Now, first we thought that might just be due to working in E. coli, something slightly different. So next we measured the transcription, what it depended on. And what we found that in E. coli, that expression of nitrogen fixation genes was dependent on poor nitrogen conditions, which you can see here in the blue line. And that was highly encouraging, because remember, this was not done under anaerobic conditions, but under aerobic conditions, where in most diazotrophs, a nitrogen fixation would not occur at all. So what we did next, we transferred our reporter system into Klebsiella oxytoca. So we work mainly with this lab strain M5A1, which we sequenced. And what we found after a few days is that NIF transcription via our reporter system also lit up under nitrogen poor conditions after a few days. Here's a control where we knocked out the transcription activator or regulatory system in Klebsiella oxytoca, and we did not see this phenomenon. What we did then, we did some confocal microscopy and looked at where in the macrocolony or biofilm, which however you want to call it, usually macrocolonies are referred to those that grow on an agarose or agar plate, whereas biofilms are those communities of bacteria that you would see in nature. So here we can see that here in the central part of macrocolony biofilm, I use these words interchangeably, I don't see too much of a difference between them. And when you look from the side, you see that inside the biofilm, we have a zone where nitrogen fixation genes are expressed. Again, here's the control. You don't see that in the control. So next we thought, wow, is that real? So we checked out if nitrogen fixation was even functional in those biofilms. And sure enough, so under nitrogen-rich conditions, we see no ethylene reduction. So here the assay is the known acetylene reduction assay, where acetylene is a mimic for dinitrogen and can be reduced by the nitrogenase. So up here, you don't see any ethylene reduction, so no nitrogenase activity, whereas under nitrogen poor conditions, you do have acetylene reduction and hence nitrogenase activity. And this, as in essence, just shows you the same thing in a bar chart. Here I want to introduce our Klebsiella strains we work with. We work exclusively with Klebsiella oxytoca. So both our lab strain and then this SGM81, which is actually from our collaborators in Gujarat University, and which has been isolated from Gujarati soil and is a super nitrogen fixer, a very, very strong plant growth promoting Klebsiella strain. So, okay, so nitrogen fixation occurs in biofilms. Now we found that about two and a half years ago, but there had been one other report on nitrogen fixation in biofilms. Sorry. And that was shown for Pseudomonas stutzeri in 2017. There are no other reports so far on nitrogen fixation occurring in biofilms. So when we look at that, at how nitrogen fixation may develop in a biofilm. You can see here it takes about 3 days for nitrogen fixation to occur in a biofilm. So once we've established that, we can grow biofilms and they fix nitrogen under nitrogen poor conditions. To look at that a little bit more in detail, what we did, we grew one of those nitrogen fixing biofilms here on the left. And then we used time-of-flight secondary iron mass spectrometry called TOF-SIMS. So basically, you ablate any kind of surface with a beam, and then you measure what ions, daughter ions, come out. And what you can see here, when you look at the depth, the estimated depth in micrometer, so you're basically drilling a hole into the biofilm on the Z-plane. And what you see that for the first 10 or so micrometer, we don't have any nitrogen fixation. The way the experiment was done, we grew those biofilms on isotopically labeled 15N nitrogen in form of ammonium, so that all molecules of the biofilms would be 15N labeled. We then transferred them and let them grow for a certain amount of time, so that the naturally occurring 14N then gets integrated into the metabolites because of nitrogen fixation. So this must come from nitrogen fixation. So here, after 10 micrometer, you see the sharp rise in ions that were 14N, indicating, here those molecules for instance, indicating that nitrogen was indeed fixed and incorporated in the biofilm matrix. So there was proof of principle that it is really occurring and that the nitrogen derives from nitrogen fixation. Now I'll come to the last part of my talk, which is which actually came from a different study we conducted in the lab, which was published a couple of years ago or so, or 3 years ago, where we looked at more general plant growth promoting traits of Klebsiella. In this case, our Indian strain, Klebsiella SGM81. And when we looked at the root hairs using confocal microscopy, the root hairs are here stained in red. We wanted to see how those plant-associative bacteria colonize the roots. And here the bacteria are GFP tagged, so they're all green. And we observed that around the root hairs, as well as the normal root, that the bacteria liked to cluster in clusters of 200-300 cells and so forth. So we thought, okay, now if we connect that, we have clustering Klebsiella on the root system, but we now know that these clusters might in fact form biofilms and could therefore reduce nitrogen to ammonium in an agricultural setting where oxygen, say, is quite high. So we did quite a few experiments just to establish the biofilms on the root system. It's sometimes successful, sometimes not. Sometimes the bacteria overgrow the plant and the plant will die. So titrating root systems with different strategies seems to be crucial. But if we try to grow biofilms on the root system, the question is, would they fix nitrogen? So what we did, we washed, once we've established a biofilm on the root system, under nitrogen poor conditions, we washed off the cells, the bacterial cells from the root, and then used an RNA fish experiment or probes that hybridize to the messenger RNA of nitrogen fixing genes. And here you have the cells we washed off under the microscope. And on the right-hand side, we used two different probes. We used one probe that reports on nitrogen assimilation, GLIN-K AMTB down here. And this probe is red. And on the other side, we used probes specific to NIF genes, in this case, the structural genes of the nitrogenase. So what we saw there, that some of the cells lit up, both with regards to nitrogen assimilation, or there were most of them that shown up for nitrogen assimilation, indicating nitrogen deficiency or starvation, but also a subset of these fixed nitrogen. So again, here we show that nitrogen fixation genes are expressed in biofilms grown on roots. Then we did similar experiment, acetylene reduction, to show the functionality of the nitrogenase in those cells that were grown on roots. And you can see that after a few days, similar to the biofilms on plates, we have acetylene reduction. We also see that in the AMTB mutant. I haven't spoken to that yet. AMTB is ammonium transporter, and which we use to excrete more ammonium to the outside, or that is at least the hope. You have some, when you look at the control, so this is just a plant with no bacteria, we also have some residual acetylene reduction. No, we have some ethylene. We detect some ethylene. And I have a question to you all. Why might that be? Does anybody have an idea why we might have ethylene? Why we detect ethylene in the gas chromatograph in plants? So this is an open question to you guys. If you have an idea. Hello? Is anybody there?
सर आंसर करना है. Actually, it was great to listen you. They can't answer you. Sorry. Sir, can you listen me? Sir, can you listen me?
Dr. George.
Sir.
Okay, Dr. George disconnected.
Disconnected.
Due to some technical issues, our speaker got disconnected. Please be connected here. He will join us soon. I hope you all enjoyed the session. He will continue us soon. Please be connected.
Dear participants, I think there is some technical problem. Dr. Schumacher is unable to rejoin it. Okay, due to some technical problem, it get disconnected. But I thought the session was wonderful. And all the things in very elaborative manner. And now we have in-depth information about how we can increase our nitrogen fixing capacity, how can analyze it, how can interpret it. So the session was really informative, and we are trying to reconnect him, but I think that there is some problem. So I think let's sum up it. We will post the feedback link, and we will try to organize again some webinar again with Dr. Schumacher in near future. Your question you can post on the chat box. We will try to send you answer for the same. We will conclude all the, we will compile all the question. We'll send to Dr. Schumacher, and we will try our best. Okay, he got connected. Okay, 1 minute.
Yes, I got disconnected.
1 minute. Dr. Schumacher. Oh, okay. We are trying. We are trying to. I think some technical problem was there.
Yes. How? I don't know,
Okay.
because I was just speaking. I just saw this my screen. How far did we get?
No problem. No problem. No problem. So now we'll conclude it. We will our some questions are there from the participants. So can you resolve it, Dr. Schumacher? Some questions from the participants.
Yes, of course.
Yeah. Ms. Ayushi, please ask the questions. Some questions from the.
Sir, first of all, I would like to thank you for acknowledging us. Now, I would like to ask our first question. Does the NIF gene cluster form operon?
Yes. In the case of Klebsiella, there's seven operons, but they're on the genome. They're clustered together, but they're differently transcribed in different strands. For example, when you look at NIF-HDK, so the structural genes of the nitrogenase, we measured the abundance of the proteins per cell, and there's a very, very strong promoter. So there we measured about 800,000 for the iron protein, which is a more abundant one, and about half of that of the MOFI protein. So there you have 800,000 copies per cell of proteins, whereas for some, they're quite low, they're in the thousands per cell. So yes, there are the promoters of the operons and have different strengths.
So next question is the approach for increasing the nitrogen assimilation same for all diazotrophs?
Pretty much, yes. Apart from the cyanobacteria, they work differently. But all proteobacteria have a similar mode, let's say, of action, if that answers your question.
So does host specificity interrupt the process?
Does host specificity, can you say that again?
Does host specificity interrupt the process?
It's a good question. It's a bit of an open question because there are a lot of studies who look at the diazotroph or bacteria in general. Since this occurs outside in the soil, you usually find in nature or in agriculture, you find a wide range of different bacterial strains that can fix nitrogen. Some people think so. For example, was it Azospirillum is perhaps happier with sugarcane, say, or others. But personally, I'm not too convinced. I think it depends on a number of factors. It depends on abiotic factors like temperature, water availability. It might depend to some degree on the plant, which bacteria they're happier with. I'm more of the opinion that the abiotic factors in favoring or disfavoring certain bacterial strains might be more important than the plant species itself. But I might be wrong on that. I cannot say. I think nobody can.
So next question is, how can we maintain the environment temperature during fixation process?
Maintain the environment. Well, bacteria and plants, they don't really have they are exposed to the temperature they are. I mean, there are, of course, some heat shock proteins as well as cold shock proteins, which allows the bacteria to fend off more extreme conditions they're unhappy with. But temperatures they're uncomfortable with. But we cannot maintain, at least in the soil, if you're thinking about that, maintain the temperature externally. I'm not sure if that answers your question.
So next question is, microbes you have taken under consideration, could they bear the temperature fluctuations of our country?
Yes, temperature is, of course, an issue. I mean, we mostly do things in the lab where we can keep the temperature constant. But it's a fair point. But I mean, Klebsiella is, for instance, happiest around 27 degrees and does the most nitrogen fixation at that temperature. But I don't think you can control the temperature in the field. You can only do that in the lab. And how that impacts on nitrogen fixation. Say you're going down to 15 degrees at night or 10 degrees and then up again during the day. It's difficult to say. But generally, one would probably say that in general, catalytic terms, usually a drop of 10 degrees reduces the general metabolic activity by about half.
The next question is, what is the effect of nitrogen-fixing bacteria on vermicompost production?
On, pardon me, what production?
Vermicompost production.
Compost production.
Yes.
Well, it depends on what kind of compost you're talking about. If it's about, usually you have then plant material, which you then compost. So initially, I would think that the compost, I don't know the compost composition, is not necessarily nitrogen poor. It probably depends on what kind of plant tissue we're talking about. So initially, I wouldn't say compost is nitrogen poor. Now, if it is, then of course nitrogen fixation could be employed there. It's a good thought. But then, given that during the decomposition of plant material, I think compost reach a reasonably high temperature. So if you were to think to add any kind of species there, you would probably want to have one that is reasonably robust with regards to temperature.
Okay, sir. Our next question is, does biofilm control bacterial diseases in plants?
That's an interesting point. It's true that biofilms in general, people know about biofilms in clinical settings, where it's obviously a bit of an issue because they get antibiotic resistance in the biofilm and so forth. I would think not. I mean, our experience is if you give them too many bacteria, if you let basically the root system be overtaken by complete coverage of thick biofilm, then it's detrimental to the plant. Because, as you know, any kind of bacteria are perceived by the plant in form of PAMs or MAMs, and therefore the plant defense mechanism comes in. So I think in soils there's a very fine balance of trying to control the titer of bacteria on the root system. So again, I would think here the trick is to balance that in a good way, in a way that's both beneficial to the plant as well as the bacteria.
Thank you so much, sir, for answering all the questions satisfactorily. And I would also thank you for such an enlightening session. I would also thank to all the participants for being attentive during the session. I would thank our HOD, Dr. Shekhar Jain, who gave us opportunity for attending this fascinating webinar. Thank you so much, sir. And now I would, yes, sir.
You're very welcome.
Thank you. And now I would request all the participants to fill the feedback form correctly. Feedback link is given in the chat box.
Dr. Jao.
Yes.
It was really a nice session. Thanks.
Welcome.
So we have so many participants, and they found it really wonderful and informative. So thanks on behalf of all the participants and of Life Science Department. It was really a nice session.
Thank you very much.
And for those participants who are asking for the video, the video will be remain available on Life Science Department channel. You can revisit it anytime and you can watch it. It is really worth watching again and again. Thanks to all participants.
Thank you very much for listening. It was a real pleasure, and thanks for the invitation again, Shikha.